Okay. So I had one of my, one student was asking about quantifying mixed urine cultures as well as urine cultures that have a low quantity. I'm not sure how well you can see this. I hope you can. It might be a little small on your screen. Once again, remember, these cultures were set up using a 10 microliter loop. So that means you're going to expect to see more growth with an ultimately small quantification. Okay. So this is a collective, this is a mixed culture. There's more than one colony type on this plate. Well, first of all, you can see the McConkey plate is negative. Okay. That gives you a lot of information. The McConkey plate is no growth. You are not growing most of your gram-negative bacilli. Okay. That means most of what you're growing, what I have on the blood agar plate, most likely is going to be gram-positive organisms. Okay. It looks to me that this is mixed. Okay. Now, if it's less than 10,000 colony forming units per mil, that means that each colony type, so not collectively, you have to look at each individual colony type that might be growing, and each one of those is less than 10,000 CFU per mil. On this plate, it means that each one of them is going to be less than 100 colonies are going of each colony type on this piece of media. Now, if it's mixed, there might be more than 100 colonies total, but because there's probably 2 or 3 organisms growing on here, each one of them is less than that threshold of reporting. Okay. So this is a mixed culture of low quantity. Now remember, if this was using a 1 microliter loop, it would be times 10. Okay. So you would actually have, instead, you'd have somewhere between 10 to the 4th and 10 to the 5th CFU per mil. Okay. Okay, let's see if I can find one. I think you probably are pretty good at getting very high quantities. Those are easier. So if it's greater than 100,000, that's pretty easy to see. It's the ones that are kind of in the middle sometimes. Here's another example of of mixed flora. Okay, at a low quantity. Okay, I'm going to try to find one that has mixed growth. Okay. So here's another colony. This is another culture that has mixed organisms on it. Now, this particular urine culture was a nephrostomy urine. That means this was a sterilely collected urine. Any amount of growth is going to be significant on these types. But let's suppose this was not a sterile urine. Okay, let's say instead this is a clean catch urine and it has more than one colony type. But you're talking about anywhere between 10, probably around 10 to 20 colonies of any type, one type of organism. So this would be, if that was the case, this would still be kind of a low quantity, not significant. Okay. Okay. So again, when you're looking at your urine cultures, if you remember, you're talking about individual colonies and the quantity. Okay. They have to fit into the primary pathogens. So if you have, for instance, a Corynebacterium, that's typically skin flora, even if it is growing a significant amount, it's not a uropathogen. Okay. So you need both the quantity and the pathogenicity of the organism. Additionally, remember, if you have more than 2 colony types growing, even if they are pathogens, and even if they are in that at least 50,000 CFU per mil, if you have 3 or more organisms, that typically is not significant in a clean catch or non-sterile urine. Okay. That typically means that you have mixed flora and it's probably contamination. Okay. All right. Okay. I'm going to really jump ahead here. Because I want to make sure this was one of the things I'm actually going to share. There we go. Okay. I'm going to go through this quantitative dilution, the susceptibility testing again. And I actually have fixed this. If you recall, this was from the PowerPoint that I shared with you earlier this year, and I found an error on it. So we're going to go through it again because I saw on the exam 4, several people got the MIC and the MBC incorrect. So I want to make sure that we understand this concept. Okay. So what I'm sharing with you, I will actually resend this to you because I did make some corrections and I highlighted some points that hopefully will make more sense to you now. Yeah. Okay. So this is the macro version of what we see in the Vitex system. Okay. That ultimately uses the same theory. Okay. And we are using serial dilutions for creating different concentrations of the antibiotic. Okay. So first concept is make sure you understand what an MIC is. Okay. The minimal inhibitory concentration. That represents the lowest antibiotic concentration that shows no visible growth. Now, if you take out this whole tube system, get rid of that no visible growth and just say it's the lowest antibiotic concentration that will inhibit the growth of an organism. Okay. Typically, this is what the physicians want to know when they're looking at how to treat an infection. They want to know how can they inhibit the growth of this organism. And typically, it's the type of antibiotic that will represent either bactericidal, which will kill the organism, or a bacteriostatic that just inhibits the organism, and the immune system will take care of the rest. Okay. Okay. So minimal inhibitory concentration versus the MBC, which represents the lowest antibiotic concentration that will kill the organism. Okay. All right. So we have a series of tubes, correct? We are going to add broth. This is uninoculated broth. This is going to be kind of our growth broth. I mean, this is the broth that we're going to ultimately going to let it incubate. But at the moment, it doesn't have anything in it. Okay. So typically, we're probably looking at maybe a Mueller-Hinton broth or a BHI, a brain heart infusion broth, something like that. Okay. So 2 through 13 have broth only. That's what we're going to start with. Okay. And tube 12 actually is going to get double the broth because that's all that's going to go in. It's not going to have bacteria. It's not going to have antibiotic. Okay. That's going to be our broth control. Next, we're going to pipette the working antibiotic solution. Okay. And we're going to start with a concentration of 200 micrograms per mil. And we're going to put a mil in tube 1. Remember, tube 1 doesn't have any broth in it. Okay. Not yet. Tube 2 has a mil of broth. And tube 13, we're going to add a mil of broth. Okay. And then nothing else is going to go into tube 13 either. Okay. Okay. So now we are going to serially dilute the antibiotic. That serial dilution, that means twofold. Every time we take 1 mil out of the more concentrated tube, the total concentration is going to be halved. Okay. So tube 2, we're going to take a mil of that and we're going to put it into tube 3, mix it. A mil of that goes into tube 4, mix it, and down the line, all the way through tube 10. Tube 10, we're going to take a mil out and we're going to dispose of it. We don't need it. After tube 10, nothing goes into 11, nothing into 12, nothing into 13, okay? Okay, here is where the mistake was made on the in the PowerPoint you received earlier this year. Okay. So at this point, now we are going to add 1 ml of inoculum, okay, into tubes 1 through 11. Okay. So this is going to be our oops. And I already see that there's an error here. It shouldn't be 200 micrograms per mil. Just ignore that, because I just want you to know that it has 1 mil of inoculum. Okay. 200 micrograms per mil doesn't, that has to do with the antibiotic. Okay. So 1 mil of inoculum goes across the board, 1 to 10 and 11, because 11 doesn't have any antibiotic. It's just going to have your inoculum control. So the problem is that in the initial PowerPoint that you were given, it didn't take into account that by adding this mil of inoculum, you've now diluted that initial concentration of antibiotic, okay? Because now we've halved it, because now we have a total volume that's double where we started, okay? So we started with 200 micrograms per mil in tube 1. Our final concentration of tube 1 is now what is been diluted 1 to 2. So now the final concentration of the antibiotic is 100, and so forth. So I've corrected each concentration of antibiotic has been corrected, okay, and halved. Once again, ignore where it says pipette 1 mil of inoculum, the 200 micrograms per mil. Just, I will take that out when I send you. You're just going to, you don't need to know the concentration of that. Just know that we've added 1 mil of the inoculum. That means we've grown the organism in a broth. Okay. Okay. This part all stays the same, except for I have corrected the concentration of antibiotic in each tube. So here is where you see tubes 1, 2, 3, 4, and 5. Okay. And then 6, 7, 8, 9, 10, 11. So 6 through 11 are obviously growing. They are visibly growing. 11 being our growth control, our inoculum control. 6 through 11 are growing. 1, 2, 3, 4, and 5 don't show any visible growth. So 5 is where our MIC is. It is the lowest concentration that is showing no visible growth. It's basically showing inhibition. Okay. It's not tube 6. Tube 6 is where you start seeing growth. Okay. So now what we do is we're going to subculture each of the tubes that look like they're not growing anything. We're going to subculture those, and we're going to incubate those plates, and we're going to look for possible growth. So in this case, tube 5, when we subcultured it, grew. Okay. Tubes 1, 2, 3, and 4, the subcultures were no growth. So they continued to inhibit and show bactericidal or bacteriostatic activity. So now tube 4 is with the 12.5 micrograms per mil of antibiotic. That is the lowest concentration that killed the organism. Okay, as our MBC. All right. So in this case, 5 is your MIC, and the concentration is 6.25 micrograms per mil. Tube 4 is how much it took to kill the organism, and it's 12.5 micrograms per mil. Okay. All right. Additionally, these 3 controls are necessary to make sure that you can you can actually call it a valid test. Okay. Okay. So I will resend this to you with the corrections so that hopefully it'll make more sense when you, go back and review that information. Okay. 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 The something else on the ASTs, the susceptibility testing I wanted to run through. I want you to make sure you review the difference between Kirby-Bauer disc diffusion testing and e-tests. They will both give you susceptibility testing, but disc diffusion will not give you an MIC because you only have one set concentration of antibiotic that is infused in that disc. You are able to tell whether, based on that concentration of antibiotic, that you have the organism is either going to be susceptible or resistant based on the standards set by the CLSI, and where you're given different ranges of the size of the zone of inhibition. Okay. However, an E-test is considered an MIC because you have a gradient of antibiotic concentration that is built into the disc. I'm sorry, into the strip, the each strip. And so as soon as that strip is placed onto the growth on the agar, it starts immediately diffusing. It's keeping it stable. That antibiotic is stable until the growth of the organism, and then it is. That's when you have the. I'm sorry, I'm losing my train of thought. That's when you have the ellipse that is formed based on that variant amount of concentration. All right, I wanted to share though Okay. This was a question that was on the virtual AST lab. And I think, and a lot of you actually did a pretty decent job with this, but I wanted to make sure we discussed it a little bit further. Okay. So it said, this is the E-test. It says, what is unusual about this E-test result of oxacillin versus Staph aureus? Okay. What are some explanations for this result? So you see, you've got an oxacillin E-test, and you can see that there seems like that there is a zone that mostly is at about 0.094 to 0.125. Okay. But there's also several colonies within that zone of inhibition in that ellipse. So the question is, what's unusual, and why do you think? What are some explanations for the results? And I got some really good answers. There were some people said, well, maybe the activity of the strip is been reduced. Maybe it's expired. Good point. That certainly could be. You could have a lack of activity using that strip, or you could. Some people said, well, maybe it was mixed. Maybe you have two different organisms that are actually growing on the plate, and so it's not a pure culture, and that's a good explanation as well. Okay. The third, and some of you did pick up on this, so I was glad to see that. Okay. Another possible explanation is that there is some kind of hetero resistance on this organism. So if you had a Staph aureus, it could be that this colony type has some mixed. It's perhaps showing some mixed resistance within the strain. Okay. And so, which is what we mean when we talk about this hetero resistance. So it could be that this organism is maybe moving towards resistance. Could be that patient might actually have two different strains of Staph aureus that are growing, and some are resistant oxacillin. So it could be there's a MRSA that is emerging. Okay, so a way to actually further test that is to isolate those colonies that are growing within that zone of inhibition, subculturing that and retesting it and seeing if you can see a more clear sign of resistance. So I just wanted to draw your attention to that because this is kind of an interesting, this was sort of an interesting test. And you guys did a pretty good job of explaining that. So I'm just glad to see that. Okay. I'm going to try to finish up here in a few minutes here. So just trying to see if there's more. Okay. I'm going to go back. Someone else asked me if we could go back and I could review some of the highlights of the units on the non-fermenting organisms, the non-fermenting gram-negative bacilli, such as Pseudomonas, and the miscellaneous gram-negative fermenting organisms and the Haemophilus organisms. So not everything, but just kind of the highlights, okay? So I'm going to pull those up here. Okay. Okay, so once again, I'm not going to go through this whole thing. Again, I'm going to make sure that you use those learning objectives. Those are a great place to start, okay? Because that's also kind of these are the objectives that also you want to be using to review as you go through towards studying for your boards as well, okay? So it's kind of it's both, okay? Some really basic information here. We're talking about these non-fermentative gram-negative bacilli is to understand most of these we're talking about their ability to utilize carbohydrates through pathways other than fermentation. So that's why they are oxidation. They're oxidase positive. These are oxidative organisms. Okay. And in general, the most common, the organism we talked about most was Pseudomonas. And the primary pathogen within Pseudomonas, the primary species in that genus is Pseudomonas aeruginosa. Okay. So these tables like this, I think, are really helpful. You know, you can't, I totally understand how you cannot memorize every single thing, every single organism, every single antibiotic, every single biochemical test. Okay. But when you think of groups of organisms, remember the basic characteristics of your Enterobacteriaceae. They will be on the left here of this table. Glucose fermentation, oxidase negative, grows on blood and McConkey. They are so they're fermentative. They're catalase positive. And these typically are very reactive in a commercial system. So they tend to have lots of carbohydrate activity, different amino acids activity, things like citrate and indole. Those are all helpful helpful biochemical tests in identifying them. Your non-fermenting organisms, not so much. They tend to be not very reactive in commercial systems. They're also mostly oxidase positive. and they don't ferment glucose. Okay. So they are, these differences are helpful, I think. Okay. There are a few, and that's what's in that other unit that we followed with like the Vibrio and such. There are some oxidase positive organisms, G and B, that are glucose fermenters, and they fall in to these genus such as Pasteurella, Aeromonas, Plesiomonas, Vibrio. Okay. That was that separate unit that we already talked about. Okay. The Pseudomonas are typically found causing opportunistic infections in patients that have been hospitalized, patients who have are on ventilators. The organisms typically really like to grow in water, and so you can see them getting into water baths and respiratory care equipment and hot tubs and things like that. Okay. Pseudomonas, one of the most significant disease processes that Pseudomonas is involved in is cystic fibrosis. Okay, very significant chronic or lung disease. Additionally, we see it in a lot of community-acquired infections such as Swimmer's ear, patients, people that get keratitis and that wear contact lenses. I would particularly pay attention to what are, here I'm going to go down to this one. What does Pseudomonas look like? What do we see? What are the most important things that we note about Pseudomonas in the laboratory? Okay. It grows well on blood, chocolate, McConkey. It's a non-lactose fermenter. It's oxidase positive. On the blood and the chocolate, sometimes you will see a sheen. It's often flat, and you'll see a sheen, kind of a dark looking organism, but it can also has a mucoid version that's particularly we look for in patients with cystic fibrosis, and it can form a pigment, a bluish green pigment that you see here on this slide on the Mueller-Hinton there. Okay, those are significant descriptions of the colony, how the colony acts. Okay, that's an important way to look at how to approach identifying this organism. Okay, I'm not going to ask you all the different varieties of biochemical tests, particularly ones that we do not typically use in the laboratory. Okay. So for instance, this exercise is helpful. These are the things you should know because this answer to this question, exercise 2, is Pseudomonas aeruginosa. Okay. But these characteristics are important in that identification. Okay. I think the most important thing you should know about Burkholderia cepacia is that it is an important pathogen in cystic fibrosis, and we can it's a slow grower, so we want to add additional selective pieces of media that inhibit the growth of organisms such as Pseudomonas, so aeruginosa, so that it doesn't overgrow. Okay, again, tables like this in the PowerPoints, I think are important. Okay. Okay. Close out of there. So in that section of oxidase positive fermenters, so remember these are organisms that are kind of similar to the non-fermenting Pseudomonas because they're non-lactose fermenters and they're oxidase positive. But these are weird in that these particular organisms are actually glucose fermenters as well. Okay, so that's why this section is these are they're kind of outside of the box. Okay, and most of the organisms within this group are not normal flora. Okay, they show up causing they typically are found in animals, and so when the humans ingest or taken water or eat contaminated food, that's when we come into contact with these organisms. The primary one I think you should know about in this section is the Vibrio and Pasteurella. Those are probably the two main ones that we've talked about. Obviously, Vibrio is known for several different organisms. Vibrio cholerae is the one that we talk about because it causes that very, very serious infection of cholera. So I would familiarize yourself or re-familiarize yourself with how we can identify Vibrio, okay? By gram stain, the special media such as the TCBS we use, and additional biochemical test, oxidase, indole positive strings test that is useful as well, okay? The two other species, Vibrio parahemolyticus, causes a GI infection. Vibrio vulnificus, we've talked about that a bunch of times, causing really serious wound infections. And because of the cytolysine present, it can lead to organ failure and death. Other organisms in here, obviously, Campylobacter is so important because it's one of the most common causes of GI infection, disease, diarrheal disease in the U.S. Okay, don't forget about several people in when we did the exam 3. That now I think it was exam 3. Yes, we're talking about stool cultures and talking about particularly exposure to poultry. They forgot about Campylobacter. Okay, they wanted to immediately go to patients that might have salmonella, which is true. That wasn't wrong, but you can't leave out suspecting Campylobacter because it actually is very common in this country. Okay, so you need to have the understanding of how Campylobacter is identified. Why is it's so different in how it's cultured from the other organisms that cause diarrheal infection. So it has to go on its own piece of selective media, incubated at 42 to 43 degrees, requires an increased CO2, decrease in oxygen, and the gram stain is also very unusual. It has that kind of curved seagull wing appearance, okay? So that part, this organism, you should refamiliarize yourself with. And already covered that. Okay. Okay, same thing, going through this unit, making sure you look at the objectives, okay? These are kind of the more, they're not even necessarily unusual, but they are other gram-negative bacilli that require special media to grow, okay? They might grow better on, for instance, they might require buffered charcoal yeast extract. That's an example of Legionella, okay? They might require Reagan low media, such as the Bordetella, okay? So from refamiliarizing yourself with the main groups of organisms in this unit, okay? Obviously, Haemophilus is one of the most important ones and most common. So how is Haemophilus, what's the gram stain look like? This organism is the main reason why we set up chocolate on sputum cultures, okay? It's to make sure we capture this very common organism that causes pneumonia, okay? If we did not set up the chocolate, you would miss this organism every time. And I'm talking about Haemophilus influenza, not the non-fastidious Haemophilus that can grow on blood and chocolate. And there are many of those. So refamiliarize yourself with how to identify and what the X and the V factor and how that aids in the identification and speciation of Haemophilus, okay? Sorry, I'm just kind of reviewing. Okay, this is just that basic first two columns here is helpful. You can see the Haemophilus influenza requires both X and V factor. Okay, that is the reason why it will only grow on chocolate. Okay, because it does require both. Haemophilus hemolyticus as well, but it's beta hemolytic. Okay. All right. I mentioned Legionella. Okay. I would review your Legionella. I would review your Bordetella, particularly pertussis. That's the organism that we are most concerned about because it causes whooping cough. And primarily due to its virulence factors that can lead to pretty significant lesions in the respiratory tract. Okay, the media that specifically for Bordetella pertussis and how we identify it. Mostly that's by PCR nowadays, but you should still know what the primary media, selective media that we use for this organism. Okay. I already told you about Pasteurella. This is important because it's very common. We see this organism quite a bit, causing animal bites or causing wound infections from animal bites or animal scratches. Okay, so know the clinical significance and the clues to identifying it. Okay. The additional biochemical tests that are necessary to identify it and that are very easy to do in the lab because these are all biochemical testing that are very accessible: oxidase, catalase, indole. The couple of the organisms that are associated with bioterrorism and biological warfare, such as Brucella and Francisella, it's a good idea to, we think we never see these, but they do show up occasionally in the lab. Francisella is the causative agent for tularemia. Again, these are organisms that are primarily infected, and the infections come from exposure from contaminated animals. Either Brucella, we see that causing infection in cattle. Francisella is from other animals such as like rabbits, sheep, other household pets that could also actually have this organism as well. Okay, they're very difficult to grow in the lab because they typically there tend to be gram-negative coccobacilli. They often don't grow well on routine pieces of media. So anytime you have a slow-growing organism, and the gram stain is a gram-negative coccobacilli, we always want to be a little cautious, okay, and look into further ways of identifying that will not cause us to be exposed to possible aerosolizing organisms that are dangerous. Okay. Also familiarize yourself with the HASEC organisms. Okay. Haemophilus, Agricobacter, Cardiobacterium, Eikenella, Kingella. Why they're important? They are rare causes of endocarditis, and but normal in the respiratory tract. Okay, so they don't grow well on McConkey or at all, and they're oxidized positive. So these organisms typically kind of looked at as a group. They're all individual organisms, but they do sort of act similarly. Okay. Okay. Like I said, I'm not going through all of these because, once again, we've already done that once. I'm just telling you kind of where I would kind of attack that area. I think I covered that. Sorry, I'm going down my little list, guys. Seeing. Make sure you go back and review your identifying your important Neisseria and Moraxella organisms, the biochemical testing that helps to speciate the significant pathogens of those organisms. The one area that is just really hard for me to summarize is going through your Enterobacteriaceae. I would start with, it's so hard because there's so many organisms. I get that. How would I approach identifying gram-negative bacilli that are in the Enterobacteriaceae family? The first thing I would do, and I did send you, I know I did email you guys some flow charts. They weren't great, but they might help a bit. And I would always start with what are the primary lactose fermenters and the primary non-lactose fermenters in the Enterobacteriaceae family. And then what are the main biochemical tests you can do? And you always are going to start with your oxidase, okay? Oxidase positive, oxidase negative. Oxidase negative organisms, Enterobacteriaceae are all should be oxidase negative, okay? Then I would look at indole, okay? See where that helps you. I would look at your urease, because once again, that's another test that you can do pretty easily in the lab. Motility. So oxidase, indole, motility, urease, those are easily performed tests in the lab and can get you pretty close to a at least a group of organisms. Okay. Also, then what are the colonies look like? Is it swarming like a proteus? Are they really mucoid like Klebsiella? Do you see hemolysis on the blood ocker? So all of those are the kinds of things that are easily observed and easily tested. Okay, so that's kind of the area that I would go to sort of get yourself down to a better identification. H2S, forgot about that. It's very, very, very important. So that's kind of how I would approach a lot of those tests. And of course, if you are good at memorization and want to look at like your IMVIC patterns and some of your other biochemical tests, fabulous. Okay. All right. One other thing I wanted to remind you of that's a really helpful thing is that work you've already done, go back through all of your weekly study questions. Okay. Those will be really helpful to you in as a review for the final as well. Okay. There was a reason why they were study questions to begin with. Okay. That meant that those were areas and questions that we thought were important to begin with. So I would use those in part of your review as well. I think I'm looking down my list. I feel like I have answered most of the questions that were put to me for review. So if you have any other specific questions that we did not cover today, go ahead and send me a message and I'll answer to the group. And if you want, you can respond. I will send you an email with a couple of these updated units, and you can respond to the group so that everyone sees your question if you want, and I can respond to the group as well. So, okay, I'm going to let you all go, and let me know if you have any more questions. Okay, good luck.